The treatment of multiple myeloma has entered the era of immuno-targeted therapy, CAR T-cell therapy, often referred to as a living drug. It is one of the most effective treatment options for multiple myeloma currently available. Fucaso equicabtagene autoleucel injection, developed by EASO Biotechnology, is the first fully human CAR T product approved globally for the treatment of relapsed and refractory multiple myeloma. Equicabtagene autoleucel injection exerts its effects by targeting B-cell maturation antigen. BCMA is considered as one of the most promising targets for the treatment of multiple myeloma. Patients' autologous T-cells are collected and sorted, transduced via non-pathogenic lentiviral vectors carrying CAR gene in vitro. Ultimately, CAR molecules are expressed on the surface of autologous T-cells. EASO Biotechnologies has achieved independent production of lentiviral vectors. The entire CAR T production process strictly adheres to GMP standards, guaranteeing product quality from vein to vein. The CAR molecule of equicabtagene autoleucel injection was selected from over 5,300 antibodies and has a fully human structure. The CAR structure consists of 5 parts: an extracellular antigen binding domain SCFV, a hinge region, a transmembrane domain, a co-stimulatory domain, and a signal transduction domain. Equicabtagene autoleucel injection's unique, fully human SCFV possesses both heavy and light chain-binding domains, which help to enhance the full coverage of the BCMA target on myeloma cells, mediating rapid activation and transduction of cellular signals. The fully human SCFV can bypass host cell immunogenicity and is less likely recognized and attacked by the human immune system, resulting in durable in vivo persistence, thereby prolonging its efficacy. Equicabtagene autoleucel injection exhibits a dual mechanism of action. On the one hand, its unique recognition mechanism breaks tumor immune tolerance, rapidly eliminating myeloma cells. On the other hand, as living cells, they can persist for a long time, effectively improving immune surveillance to prevent myeloma relapse. In the immune system of a healthy adult, T-cell receptors recognize antigen peptides presented by antigen-presenting cells through the major histocompatibility complex. With the assistance of co-stimulatory signals, T-cells are fully activated and exert their functions. However, in the MM patient immune system, the downregulation of MHC expression disrupts immune surveillance, leading to immune evasion and disease progression. Equicabtagene autoleucel injection delivers genetically modified autologous T-cells carrying the CAR structure to directly recognize tumor cells bypassing the MHC presentation mechanism, avoiding the escape mechanism of tumor cells and breaking the host immune tolerance state. Activated CAR T-cells secrete granzyme and perforin, directly leading to rapid apoptosis of multiple myeloma cells. At the same time, CAR T-cells can induce the death receptor pathway synergistically with cytolytic degranulation, promoting the apoptosis of multiple myeloma cells. Under the co-stimulatory effect of antigen signal activation and the second signal, CAR T-cells achieve multiple rounds of persistent expansion. BCMA on the surface of myeloma cells is cleaved by gamma secretase, forming soluble BCMA. High levels of soluble BCMA not only indicate a high tumor burden, but also affect the treatment of anti-BCMA drugs. The function of the CAR molecule in equicabtagene autoleucel injection is not affected by soluble BCMA. Rapid and deep remission can also be achieved in a high tumor burden environment. On the other hand, the cells from equicabtagene autoleucel injection can persist in vivo for a long time and effectively improve immune surveillance. First, CAR T cells with specific tumor antigen recognition activity can persist in the body for a long time and thus achieve immune surveillance of multiple myeloma cells. At the same time, The cells from equicaptogene autoleucel injection can differentiate into memory T cells in the body, achieving rapid immune response and reducing the risk of recurrence of multiple myeloma cells. Interleukin-2 and a variety of other cytokines secreted during the expansion and differentiation of CAR T cells from equicaptogene autoleucel injection help regulate the functions of immune cells and cellular immunity and stimulate host T cells, which can achieve long-term anti-tumor effects. The dual mechanism of equicaptogene autoleucel injection is breaking through the therapeutic dilemma of multiple myeloma, offering hope for a cure to patients with relapsed or refractory multiple myeloma.